If I have to remind you a topic that we are currently discussing, we are on topic number 6, and we were discussing about antigen presentation and T lymphocyte and their function and the function of the major histocompatibility complex molecule. So we'll continue in this part of processing of antigen and finish up with the presentation of non-protein antigen to T cell. So let us continue. Processing of protein antigen. The pathway for antigen processing conveyed protein antigen. The pathway for antigen processing conveyed protein antigen present in the cytosol or internalized from the extracellular environment into a peptide and load these peptide onto MHC molecules for display to T lymphocytes. The mechanism of antigen processing have evolved to generate peptides that have the structure characteristics required for the associating with the MHC molecules, and to place these peptides in the same cellular location as newly synthesized MHC molecule, MHC protein with available peptide binding cleft. Peptide binding to MHC molecule occur before cell surface expression, and it is an integral component of biosynthesis and assembly of MHC molecules. In fact, as mentioned earlier, peptide association is required for the assembly of on-surface expression of the stable class 1 and class 2 MHC molecule. Proteins that are present in the cytosol are degraded by proteosomes to yield a peptide that are displayed on class 1 MHC molecules, whereas the proteins that are ingested from extracellular environment and are sequenced in vesicle are degraded in lysosome or later endosomes to generate a peptide that are presented to class 2 MHC molecules. See the figures we'll see later. In the process of cross-presentation described later, antigen are ingested into a vesicle and then transported into a cytosol where they are processed for display by class 1 molecules. Thus, the site for proteolysis is the key determinant of MHC molecule class 1 or class 2 to which generated peptide will bind. As we have discussed previously, the function of CD8 T cell, cytotoxic T cell, is to kill cell producing foreign antigen in the cytosol, and function of CD4 T cell is to activate macrophage and B cells, which may have ingested the microbes and protein antigens. The pathway for antigen processing plays a role in the pathway for antigen processing plays a key role in determining the types of microbes and protein antigen that these classes of T cells recognizes and to which they respond. We first describe these two pathways of antigen processing and then their functional significance. So class 1 MHC pathway for processing and presentation of cytosolic proteins. The sequence of events in antigen presentation on class 1 MHC molecule is illustrated in figure 6.1.4, and the individual steps are described next. Source of protein antigen degraded in proteosomes. Microbial protein present in cytosol that undergo proteasomal degradation are derived from microbes that either produce antigen in the cytosol of cell or whose antigen are transferred to the cytosol. The same principle applied to tumor antigen. These cytosolic antigen come from the number of sources. So this is the pathway for antigen processing and presentation in class 1 MHC pathway. Top panel, protein antigen in the cytosol are processed by proteosomes. This is the top panel, you see. And the peptides are transported to the endothelial reticulum where they bind to class 1 MHC molecule. In class 2 MHC pathway, the bottom panel, this one, proteins antigen that are degraded in lysosomes bind to class 2 MHC molecule detail of these processing pathway are shown in figure 6.14 and 6.115. Cytosol, these are just the definition, the plasmic reticulum and other. So here for class 1, what happened is there is cytosolic protein. This one is digestion of protein in proteosomes, peptide in cytosol. These are broken down now. We see they are small now. They are put in the groove of they pass in they attach in MHC, then they pass through they pass through the tap. They attach with MHC class 1. Then after attaching, they move from the endoplasmic reticulum, ready to be presented outside, and CD8 T cell will see the peptide which have been binded. For this one, these are the intracellular organisms. Most of them, as you see, it is already in the cell. These for extracellular organisms which are taken by endocytosis of extracellular protein. Now digestion of protein is done by endosomes or lysosomes instead of digestion of proteases. So what happened then? This one is broken down. Then the invariant chain in endoplasmic reticulum is released. When it is released, this invariant chain is combined with part of endoplasmic reticulum. Then it combined with broken down proteins. Then they fuse together. After fusing, then they are presented outside to CD4 T cells. This is the pathway that's happened. So comparison features of class 1 and class 2 major histocompatibility complex pathway of the antigen processing and presentation. So feature, class 1 pathway, class 2 pathway. So feature, composition of a stable peptide MHC complex. Here the pathway for class 1 is peptide. This is alpha beta microglobulin this is actually MHC. Polymorphic chain of beta microglobulin peptide. So here for composition stable peptide is peptide, alpha and beta, polymorphic alpha and beta chain peptide. So here is, you see this is beta 2, this is just alpha and beta chains, peptide and peptide. Types of antigen presenting cells here, all nucleated cells. And here they are very specific. Only those professional cells they can do this. The dendritic cell, the mononuclear phagocytes, B lymphocytes, endothelial cells, and thymic epithelial cells. These are the ones that are able to present. Response T cells, site of antigen for CD4 cells, it is proteosomes. For CD4 T cells, it's a late endosomal and also lysosome. Source of protein antigen. So here mainly for CD8 T cell, the main source of antigen is mainly cytosolic proteins, usually synthesized in the cell, may enter cytosol from the phagosomes, also nuclear and membrane protein. Here for the CD4 T cell, the source of antigen are endosomal and lysosomal protein, mostly internalized from extracellular environment. Enzymes responsible for protein degradation for MHC class for MHC class 1. Endosomes responsible is enzyme responsible is beta 1, beta 2, and beta 5 subunit of proteosomes. While in class 2 in endosomal and lysosomal protease, this is a cathepsin. The site of peptide loading MHC, it is in MHC class 1 is endoplasmic reticulum, that's in. Here for MHC class 2, it is late endosomal, lysosomal. So here you remember it was detached, one of the part of from reticular endothelium was detached with MHC, then it combined together with peptide protein. Molecular involved in transport of peptide and the loading of MHC molecules. Here TAP, it's always TAP 1 and TAP 2, so TAPsin. Here invariant chain and DM. These are responsible. So what is DM? The DM is actually not defined here, but for TAP is transporter associated with antigen processing. So actually, when we go back and we find ourselves the class 1 pathway, you remember sources of protein antigen degraded in proteosomes. We are discussing that was the main thing. Actually, we mentioned about microbial protein present in cytosol that undergo proteosomal degradation are derived from microbes that either produce antigen in the cytosol of the cell or whose antigen are transferred to the cytosol. Some principles apply to tumor antigen. These cytosol antigen come from number of sources. These are cytosol antigen. They are coming from number of sources. So here we are going to see them. Number one, all viruses replicate and survive in infected cell and thus synthesize protein in the infected cell cytoplasm. So here again you get the protein. These are the most common type of microbial protein that are processed by proteosomes and presented on a class 1 MHC molecules. The MHC peptide complex are then recognized by differentiated functional cytotoxic lymphocytes and infected cells are killed. So that's number one. Number two, some bacteria are internalized into phagosomes but are able to damage phagosomes membrane and create pore through which the microbes and their antigen enter the cytosol. Again, for this one, because they have destroyed the phagosome, which is responsible for MHC class 2, now because they're in the cytosol, for instance, pathogenic strain of Listeria monocytogenes produce a protein called listeriolysin and enable bacteria to escape from vesicle into the cytosol. This escape is a mechanism that bacteria may have developed to resist the killing of microbiocidal mechanism of phagocyte, most of which are concentrated in phagolysosomes. Once the antigen of phagocytes microbes are in cytosol, they are processed in a proteasome like other cytosolic antigen, therefore presented to MHC class 1. Some bacteria have type 3 secretion system that ingest bacteria protein into the cytosol. Numerous pathogens including Yersinia pestis, Salmonella typhi, Shigella dysentery, Vibrio cholera, and Chlamydia species inject signaling protein into the host cytosol to manipulate host function and immunity to pathogen advantage. This is the major mechanism mechanism of bacterial virulence. The other factor is the other source of protein is the product of mutated gene in tumor produce antigen in cytosol for the tumor cells. As in virus infected cells display this tumor antigen or class 1 MHC molecule enables class 1 MHC molecule enables the differentiated cytotoxic T cell to kill the tumor. Initiating immune response against viruses and tumor require antigen capture of dendritic cell and transport of antigen-bearing dendritic cell to secondary lymphoid organ, there where the antigen can be presented to naive CD T cell. So initiative of immune response against the virus and tumor require antigen capture by dendritic cell and the transport of antigen bearing dendritic cell to lymphoid organ where they are presented to CD8 T cell, the naive ones. But most viruses infect cell other than dendritic cell, and tumor antigen cell are produced in tumor cell, not in dendritic cell. The process by which antigen of other cells, virus infected or tumor cells, are presented by dendritic cell is called cross presentation or cross priming, to indicate that one cell type, the dendritic cell, can present antigen from another cell, the virus infected or tumor cell, and prime or activate T cell specific for those antigen. In this process, specialized type 1 conventional dendritic cell or other antigen presenting cell capture infected or tumor cell on their antigen into vesicle. This vesicle fused with the endoplasmic reticulum, and the mechanism that remained poorly defined. Protein from the vesicles are transported into cytosol. So if transported to the cytosol, what happened? It is killed by MHC class 1. Here we are. The production of protein in delivered to cytosol. You see, maybe the viral protein transported in nuclear, then this one viral infection has happened. You see, this is synthesized viral protein. These viral proteins, they can be cross presented to maybe we see this one is already here. So the virus infected tumor cell is taken by this ingested of virus infected or cell or protein from either. So it is taken, then it is phagocytosed, then it transported the ingested protein into cytosol. Then from cytosol is broken down by proteosomes, then peptides are released. When peptides are released, they will go through TAP. Through TAP, they will go out and they will find, you remember the endoplasmic reticulum has the MHC class 1. So they will bind together and move together, and extracellular vesicles then are presented to CD8. So class 1 major histocompatibility complex pathway of antigen presentation. The step in the processing of cytosol protein are described in the text. The protein may be produced in cytosol of an infected cell or tumor cell, not shown. Other proteins that are ingested into the vesicle may be translocated into the cytosol and processed like cytosolic protein, known as to cross presentation. Beta 2M, beta 2 2 microglobulin E are endoplasmic reticulum associated peptide and the endoplasmic reticulum together with TAP transporter acetylene. UB is ubiquitin. So this is actually the cross presentation. This is the production of protein in or derival to the cytosol. Then proteolytic degradation is happening. Then peptide transport from cytosol to endoplasmic reticulum, where they will meet to the MHC class 1. Then assembly of peptide class 1 complexes in endoplasmic reticulum. Thereafter, we see the surface expression of peptide class 1 complexes and the presentation. In addition to this microbial antigen, protein produced in endoplasmic reticulum that either do not fold properly or fail to assemble correctly in this compartment are translocated out of endoplasmic reticulum and are degraded in the proteosomes. Some nucleus proteins also are degraded in the protosome. These types of protein are often found in damaged cell and tumor and may be involved in T cell mediated elimination of these cells. Digestion of protein in proteosomes. Cytosolic protein are degraded in proteosomes to generate peptides that are able to bind to class 1 MHC molecule. Proteosomes are large multiproteins enzymes complexes with a broad range of proteolytic activity that are found in a cytoplasmic and nucleic nuclei of the most cell. A proteosomes appear as a cylindrical composed of the stacked array of two inner beta rings and two outer alpha rings, each rings being composed of 7 subunits which are cup-like structure at which each of the cylinder with a capsule structure at each end of the cylinder. The protein in the outer alpha ring are structural and lack proteolytic activity. In the beta ring, 3 of the 7 subunits, beta 1, beta 2, and beta 5, are catalytic site for proteolysis. Proteosomes perform a basic housekeeping function in the cell by degrading many damaged or improperly folded protein. Protein synthesis normally occurs at a rapid rate, with about 6 to 8 amino acid residues being incorporated into elongating polypeptide chain every second. This process is error prone, and it is estimated that approximately 20% of newly synthesized protein are misfolded. This newly translated but defective peptide, as well as protein that are damaged by cellular stresses, are targeted for proteosomal degradation by covalent linkage of several copies of a small peptide called ubiquitin. Protein with chain of 4 or more ubiquitin are recognized by proteosomal cap and then are unfolded. The ubiquitin is removed and the protein are threaded through the proteasome where they are degraded to peptide. The proteasomes have broad substrate specificity and can generate a wide variety of peptide from the cytosolic proteins, but usually does not degrade them completely into single amino acid. The composition of, proteosomes influences the peptide that are produced. Proteosomes are organelles whose a basic cellular function has been adapted for a role in antigen presentation. There are two types of proteosomes with specialized function in immune system. Immunoproteosomes are present in immune cells, such as dendritic cell and other antigen-presenting cell. They contain three unique catalytic subunits known as B1I, B2I, and B5I in the B ring. The expression of the three subunit is also increased in response to interferons produced in innate and adaptive immune response. The production of these subunits results in the changes in substrate specificity of the proteasome such that the peptide produced usually contain a carboxyl terminal hydrophobic amino acids such as leucine, valine, isoleucine, and methionine, or a basic residue such as leucine and arginine. This kind of C-termin are typical of peptides that bind with high affinity to class 1 molecule. Thus, immunoproteosis plays an important role in generating peptide from foreign protein that stimulate the CD8 T cell. The second type of proteasome is called the Thymo Proteosomes because it is present in thymic endoepithelial cell. It contains a unique subunit called beta 5T, which confer upon it the ability to produce a peptide that bind weakly to class 1 MHC molecules. As we will see in chapter 8, in thymus, these peptides are derived from self-protein and their low affinity recognition is important for the process of positive selection, which preserve maturing T cell that strongly recognize foreign antigen. In absence of the beta 5T unit, e.g. in mice in which the gene is deleted, the CD8 T cell fail to mature. Predictably, CD8 cells are not affected because we will see later the peptides that are recognized by CD4 cells are not generated in the proteasomes. Transport of peptide from cytosols to endoplasmic reticulum. Peptide generated by proteasomes in cytosol are translocated by a specialized transporter into an endoplasmic reticulum where new synthesized class 1 MHC molecules are available to bind the peptide. This delivery is mediated by a diameter protein located in the endoplasmic membrane called a transporter associated with an antigen processing TAP, which is the member of one ABC transporter family of the protein, many of which mediate antigen ATP-dependent transport of low molecular weight compound across the cellular membrane. Although the TAP heterodimer has a broad range of specificities, it optimally transports peptide ranging from 8 to 16 amino acid in length and containing carboxyl termin that are basic or hydrophobic. As mentioned earlier, these are the characteristics of peptides that are generated in the proteasomes and are able to bind to class 1 MHC molecule. Assembly of peptide class 1 MHC complex in endoplasmic reticulum. Peptide translocated in the endoplasmic reticulum bind to a newly synthesized class 1 MHC molecule that are associated with the TAP dimer through tapsin. On the luminous side of endoplasmic membrane, the TAP protein associated with the protein called tapsin, which also has affinity for newly synthesized empty class 1 MHC molecule. The tapsin is part of the peptide loading complex, the outer protein in this complex being a thiol oxoreductase called ER endoplasmic electron P57 that can break and remake the sulfide bond in protein and an ER luminochaperone called the reticulin. Within this complex, trypsin form a stable disulfide bonded heterodimer with ERP55 and bring the TAP transporter adjacent to the class 1 molecule that are awaiting the arrival of peptide. Synthesis and assembly of class 1 MHC molecule involve a multi-step process in which peptide binding play a role. Class 1 alpha chain and beta 2 microglobulin are synthesized in the endoplasmic reticulum. Appropriate folding of nascent alpha chain is assisted by chaperone proteins such as membrane chaperone calnex. Within the endoplasmic reticulum, the newly formed empty class 1 MHC dimer remain linked to peptide loading complex. Peptides that enter the endoplasmic reticulum through a tap and peptide produced in endoplasmic reticulum, such as a signal peptide from membrane or secretory protein, are often trimmed to appropriate size for MHC binding by endoplasmically associated aminopeptides. The peptide is then able to bind the cleft of adjacent class 1 molecule. Class 1 MHC molecule. The peptide loading complex not only deliver peptide to newly synthesized class 1 MHC molecule, but also select peptide that bind with highest affinity to class 1 MHC molecule, preferentially over the low affinity binding peptide. This is a quality control mechanism in antigen processing. Once class 1 MHC molecule are loaded with peptide, they no longer have affinity for trypsin. So the peptide loaded class 1 MHC molecule are released and are able to exit the endoplasmic reticulum and be transported to the cell surface. In the absence of bound peptide, many of the newly formed alpha chain beta microglobulin dimer are unstable and cannot be transported efficiently from endoplasmic reticulum to Golgi complex. These misfolded empty class 1 MHC complexes are transported into the cytosol and eliminated by proteosomal digestion. This process is called endoplasmic reticulum associated degradation, but the actual degradation occurs in proteosomes in the cytosol. Peptide transported into the endoplasmic reticulum preferentially bind to class 1 but not class 2 MHC molecule for two reasons. First, the newly synthesized class 1 MHC molecules are attached to luminal aspect of peptide loading complex, and they capture peptide rapidly as the peptide are transported into endoplasmic reticulum by tap. Second, as discussed later, the peptide binding cleft of newly synthesized class 2 molecule in the endoplasmic reticulum are blocked by a protein called invariant chain. Surface expression of peptide class 1 MHC complexes. Class 1 MHC molecule with a bound peptide are structurally stable and are expressed on cell surface. The stable peptide class 1 MHC complexes that were produced in the endoplasmic reticulum are guided by chaperones to move through the Golgi complex and are transported to the cell surface in exocytic vesicle. Once expressed on the cell surface, the peptide class 1 complex may be recognized by peptide antigen specific CD8 T cell with CD8 co-receptor playing an essential role of binding to non-polymorphic region of class 1 MHC molecule. Several viruses and tumor have evolved mechanisms that interfere with class 1 assembly and peptide loading, emphasizing the importance of this pathway for antiviral and anti-tumor immunity. Class 1 MHC pathway for presentation of protein degraded in acidic vesicle. Class 2 MHC pathway for presentation of protein degraded in acidic vesicle. Generation of class 2 MHC associated peptide from endocytosed antigen involve the proteolytic degradation of internalized protein in late endosomes and lysosomes, and the binding of the peptide to class 1 MHC class 2 MHC molecule in this acidic vesicular compartment. This sequence of events is illustrated in figure 6.15, and individual steps are described next. Ingestion of protein antigen into vesicle. Most class 2 MHC associated peptides are derived from the protein antigen that are ingested into and digested in endosome and lysosomes in APC, antigen presenting cell. Proteins that are ingested into the vesicle are mostly commonly extracellular protein captured by endocytosis, pycnocytosis, and phagocytosis, but also include cell surface protein that are being endocytosed and degraded, and intracellular protein that may be membrane bound, vesicular, or cytotoxic that are included in phagocytosis during the process of autophagy. So here there is a uptake of extracellular protein into the vesicular compartment of antigen presenting cell. Then this is taken, as we are told, the endocytosis, pycnocytosis, or any phagocytosis. Then this one, when it goes in, we have the endosomes that have been taken, then they combine with the lysosomes. Then when they combine with the lysosome plus endosome, and there is a breaking down up of the proteins to become small one, the peptide. Then the chaperones, alpha and beta, this one, I think there is information that is transported here. The MHC class 2 will move out of endoplasmic reticulum, go to Golgi apparatus. From Golgi apparatus, the exocytic vesicle is moving out. Then exocytic vesicle, when it's moving out, it will come and fuse with something that is coming from this angle. When they fuse, now the MHC class 2 and the proteins are binding together, and they can present themselves to CD4 T cell. So the class 2 major histocompatibility complex pathway for antigen presentation. The stage in the process of extracellular antigen are described in the text, as you will see later. Different antigen presenting can bind native protein antigen in several ways and with varying efficiency and specificities. So the dendritic cell and macrophage expresses variety of surface receptors such as a lectin that recognize a structure shared by many microbes. These antigen-presenting cells use the receptor to bind and internalize efficiently. Macrophage also expresses a receptor for Fc portion of the antibodies and receptor for complement protein of C3b, which bind antigens that are opsonized by antibodies or complement protein and enhance antigen internalization. Another example of specific receptor on antigen presenting cell is surface Ig on B cell, which, because of its high affinity for antigens, they can effectively mediate the internalization of the protein present at very low concentration in the extracellular fluid. After the bound protein antigen are internalized, they become localized in intracellular membrane bound vesicle called endosomes. The endosome pathway of intracellular protein traffic communicate with the lysosomes, which are denser membrane bound enzymes containing vesicles. Particulate microbes are internalized into vesicle called phagosome, which may fuse with the lysosome, producing vesicle called phagocytosis. So microbes such as Mycobacterium and Leishmania may survive and even replicate within phagosome or endosome, providing a persistent source of antigen in vesicle compartment. Protein other than those ingested from extracellular milling can also enter the class 2 pathway. So some protein molecule designated for secretion may end up in the same vesicle as class 2 MHC molecule and may be processed instead of being secreted. Cytoplasmic and membrane protein may be processed and displayed by class 2 MHC molecule. In some cases, this may result from the enzymatic digestion of cytoplasmic content, the process known as autophagy. In this pathway, cytosolic protein are trapped within a membrane-bound vesicle called autophagosomes, which fuse with lysosomes, and cytoplasmic protein are proteolytically degraded. The peptide generated by this route may be delivered to the same vesicle compartment as the peptide derived from ingested antigen. Autophagy is primarily the mechanism of degrading cellular protein and recycling their product as source of nutrient during time of stress. It is also participate in the destruction of intracellular microbes which are enclosed in vesicle and delivered to lysosome. Some peptides that associate with the class 2 MHC molecule are derived from membrane protein that may be recycled into the same endocytic pathway as extracellular proteins. Thus, even viruses which assemble in cytoplasmic of infected cell may produce protein that are degraded into the peptide that enter the MHC class 2 pathway of antigen presentation. This may be a mechanism for activation of viral antigen specific CD4 helper T cell. Proteolytic digestion of antigen in acidic vesicles. Internalized protein are degraded enzymatically in the endosomes and lysosome to generate a peptide that are able to bind to the peptide binding cleft of the class 2 MHC molecule. The degradation of the protein antigen in vesicle is mediated by proteases that have acidic pH optimum. So the most abundant proteases of late endosoma are capsaicin, which are thiol and the aspartame proteases with a broad substrate specificities. Several capsaicin contribute to the generation of peptide for the class 2 pathway. Partially degraded or cleaved proteins bind to the open-ended cleft of class 2 MHC molecules and are then trimmed enzymatically to their final size. Biosynthesis and transport of class 2 MHC molecule to endosomes. Class 2 MHC molecules are synthesized in endoplasmic reticulum and transported to endosomes with an associated protein, the invariant chain, which occupy the peptide binding cleft for newly synthesized class 2 MHC molecule. The alpha and beta chains of class 2 MHC molecules are coordinately synthesized and associate with each other in the endoplasmic reticulum. The folding and assembly of class 2 MHC molecules are aided by endoplasmic resident chaperones such as calnexin. The invariant chain associated with the class 2 MHC dimer in the endoplasmic reticulum and direct newly formed class 2 MHC molecule from a transgold to late endosomes and lysosome where internalized protein have been proteolytically degraded into peptide. It also prevents the class 2 molecule from traveling to the cell surface. The invariant chain is the trimer composed of 30 Kd subunits, each of which bind one newly synthesized class 2 MHC alpha beta heterodimer in a way that block the peptide binding cleft and prevent it from accepting the peptide. As a result, class 2 MHC molecule cannot bind and present peptide they encounter in endoplasmic reticulum, leaving such peptide to associate with the class 1 molecules described earlier. The class 2 MHC molecules are transported in vesicle from endoplasmic reticulum to Golgi. Vesicle binding from the transported Golgi that contain class 2 MHC invariant complex are transported to the lysosome. Thus, class 2 MHC molecule encounter antigen peptide that have been generated by proteolysis of endocytosed protein in lysosomes, and the peptide MHC association occur in these vesicles. So association of processed peptide with class 2 MHC molecules in vesicle. Within endosome or lysosomal vesicle, the invariant dissociate from class 2 MHC molecule by a combined action of proteolytic enzyme and HLA-DM molecule and peptide derived from the protein antigen are then able to bind to available peptide binding cleft of the class 2 MHC molecule. Although a class 2 MHC molecule are relatively resistant to lysosomal protease, the invariant is degraded in this compartment. The same proteolytic enzyme that generates the peptide from internalized proteins such as caspecin also act on the invariant, leaving only 24 amino acid remnant called the class 2 associated invariant chain peptide CLIP, which sit in the peptide binding cleft. Enzymic degradation of transmembrane portion and cytosolic tail of invariant chain prevent tethering of class 2 MHC molecule to the cytosol, to the lysosome membrane, and this allows class 2 MHC peptide complex and some radio MHC class 2 clip complexes to bud out of acid degradatory vesicles and go to the cell surface. So, synthesis of class 2 MHC in endoplasmic reticulum. So you see it is moving, but it is having the invariant chain. So this one transported MHC with an invariant vesicles. Then it move and it is budded off. So it comes in proteolytic degradation of invariant chain is degraded. Then because we have HLA DMI catalyzed clip, then HLA the peptide is bound here with this one, then it is presented. So HLA-DM molecule edit the repertoire of peptide being presented, favoring the display of peptide that bind with high affinity to class 2 MHC molecule. This displacement of CLIP on its replacement by high affinity antigen peptide in lysosome are accomplished by action of molecule called HLA-DM, also called DM or H2M in mouse, which is encoded within the MHC and has a structure similar to that class 2 MHC molecule and co-localize with the class 2 MHC molecule in certain endosome. Unlike class 2 MHC molecule, DM molecule are not polymorphic and they are not expressed on cell surface. DM bind to the beta chains of class 2 MHC molecule in a region where this chain forms a peptide cleft and dislodges loosely bound peptide from the cleft. It thus acts as peptide exchanger, facilitating the removal of cleft and the addition of higher affinity peptide derived from protein antigen to class 2 MHC molecule. Peptide that bind to MHC molecule with high affinity cannot be displaced by DM. Thus, the presence of DM is important for selecting peptide that bind strongly to MHC molecule in each individual and displaying these peptides to T cell. Another dimeric class 2 MHC like molecule called HLA-DO bind to HLA-DM in lysosome and negatively regulate the function of DM. DM can only mediate a peptide exchange after being released from DO in response to cytokines and other stimuli produced during infection. DM level rise, but DO level do not, resulting in more efficient peptide exchange and antigen presentation. HLA-DO function like a chaperone for HLA-DM. Because the end of the class 2 MHC peptide binding cleft are open, large peptide may bind and then it timed by proteolytic enzyme to appropriate size for MHC recognition. As a result, the peptides that are actually presented attached to the cell surface class 2 MHC molecule are usually 10 to 30 amino acid long and typically have been generated by this trimming process. Expression of peptide class 2 MHC complexes on cell surface. Class 2 MHC molecules are stabilized by bound peptide, and stable peptide class 2 complexes are derived to the surface of the antigen presenting cell, where they are displayed for recognition by a CD4 plus T cell. The transport of class 2 MHC peptide complexes to the cell surface is thought to occur by fusion of vesicular tubular extension from lysosome to plasma membrane, resulting in the delivery of loaded class 2 MHC complexes to the surface.
Once expressed on antigen-presenting cell, the peptide class II complexes are recognized by peptide antigen specific CD4 T cell, with the CD4 co-receptor playing an essential role for binding to the non-polymorphic region of the class II MHC molecule. Surface class II MHC peptide level are regulated by modulation of class II MHC degradation. Class II MHC molecules are normally recycled and degraded by ubiquitin proteosomes system. A ubiquitin E3 ligase called MACH1 recognize the tail of the class II MHC molecule and target them for degradation. In response to the microbes, the cytokine produced during infection, antigen-presenting cell shut off the expression of MACH1 and thus increase the amount of relevant class II peptide complexes on cell surface. Physiological significance of MHC-associated antigen presentation. So far, we have discussed the specificity of CD4 and CD8 T lymphocytes for MHC-associated foreign protein antigen and the mechanism by which complexes of peptide and MHC molecules are produced. In this section, we will consider how the central role of MHC in antigen presentation influences the nature of T cell response to different antigen and the type of antigen that T cell recognize. Nature of T cell response. The presentation of cytosolic versus vesicular pathway by class 1 or class 2 MHC pathway respectively determine which subset of T cell will recognize the antigen found in these two pools of proteins and immediately linked to the function of T cell. Endogenously synthesized antigens such as viral and tumor protein are located in cytosol and are recognized by class 1 MHC restricted CD8 cytotoxic lymphocyte cell, which kill the cell producing the intracellular antigen. Conversely, extracellular antigen usually end up in endosomal vesicles and activate class 2 MHC restricted CD4 T cell because vesicle proteins are processed into class 2 binding peptide. CD4 T cells function as helper to stimulate B cell to produce antibodies and activate macrophage to enhance their phagocytic function. But both mechanisms that serve to eliminate extracellular antigens. Thus, antigen from microbes that reside in different cellular locations selectively elicit the T response that are most effective at eliminating that type of microbes. This is especially important because the antigen receptor of cytotoxic T lymphocyte and helper T cells cannot distinguish between extracellular and intracellular microbes. By segregating peptide derived from these types of microbes, the MHC molecule guides CD4 and CD8 subsets of T cell to respond to microbes that each subset can best combat. Immunogenesis of protein antigen. MHC molecule determine the immunogenicity of protein antigen in two related ways. First, the epitope of complex protein that elicit the strongest T cell response are the peptides that are generated by proteolysis in antigen presenting cell and bind most avidly to MHC molecule. If an individual is immunized with a protein antigen, in many instances, the majority of the responding T cells are specific for only one or few linear amino acid sequence of the antigen. These are called the immunodominant epitope or determinants. The proteins involved in antigen processing produce a variety of peptides from natural protein, and only some of these peptides possess a characteristic that enable them to bind to MHC molecule present in each individual. It is important to define a structural basis of immunodominance because This may permit the efficient manipulation of the immune system with the synthetic peptide. An application of such knowledge is the design of vaccine. For example, a viral protein could be analyzed for presence of amino acid sequence that would form a typical immunodominant epitope called capable of binding to the MHC molecule with high affinity. Such analysis can be done experimentally or in silico. Synthetic peptide containing these epitope may be effective vaccine for eliciting T response against the viral peptide expressed in an infected cell. Similarly, peptide produced by mutated genes in cancer are analyzed for their ability to bind to the class 1 molecule in each patient with cancer. The ones that bind are most likely to stimulate the antitumor immunity in that patient. So there is antigen uptake class 1 pathway, cytosolic antigen, this one CD8 killing experienced. Here macrophages, they present extracellular microbes, the microbes in endosomes. CD4 cytokine, they produce cytokine which macrophage activation killing of phagocytosis can occur. Otherwise, antigen B cell, extracellular antigen you see. Then this one cytokine are produced the B cell antibody secretion, antibody binding to antigen happen. So the second of two related way in immunogenesis of proteins is the expression of particular class 2 MHC allele in an individual determining the ability of that individual to respond to particular antigen. As discussed earlier, the IR gene that control antibody response are class 2 MHC genes. They influence immune responsiveness because various class 2 molecules produced by different alleles differ in their ability to bind different antigenic peptide and therefore stimulate specific helper T cell. The causes of inheriting a given MHC allele depend on the nature of peptide antigen that can bind the MHC molecule encoded by that allele. For example, if the antigen is a peptide from ragweed pollen, the individual who expresses a class 2 molecule capable of binding the peptide would be genetically prone to allergic reaction against pollen. Conversely, some individuals do not respond to vaccine, such as hepatitis B virus surface antigen vaccine, presumably because HLA molecule cannot bind and display a major peptides of vaccine antigen. Presentation of non-protein antigen to T cell. T cell also recognize and react against small molecule and even metal ions in an MHC restricted manner. In fact, exposure to some small molecule that are used as therapeutic drug to metal, such as nickel and beryllium, often lead to the pathological T cell reaction called the hypersensitivity reaction. There are several ways in which this non-peptide antigen may be recognized by MHC-restricted CD4 and CD8 T cell. Some of the chemicals are thought to covertly modify the self-peptide or the MHC molecule themselves, creating an altered molecule that are recognized as foreign. Other chemical bind non-covalently to MHC molecules and alter the structure of the peptide binding cleft such that MHC molecule can display peptides that are not normally presented, and these peptide MHC complexes are seen as foreign. Several small populations of T cells other than CD4 and CD8 cells are able to recognize non-protein antigen without involvement of class 1 and class 2 molecule. These population of exception to the rule that T cell can see only MHC associated peptide. The best defined of these population are natural killer cell T, natural killer T cells, and the alpha delta T cell. The natural killer T cell expresses the marker that are characteristics of both natural killer cell and T lymphocyte and expresses the alpha beta T cell receptor with very limited diversity. The natural killer T cell recognize lipid and glycolipid displayed by class 1 MHC like molecule called CD1. There are several CD proteins expressed in human and mice. Although their intracellular traffic pathway differ in subways, all CD1 molecules bind and display a lipid by unique mechanism. Newly synthesized CD1 molecule pick up cellular lipid and carry these to the surface. From here, CD1 lipid complexes are internalized into the endosomes and all lysosomes, where lipid can then be ingested from external environment, where lipid that have been ingested from the external environment are captured and new CD1 lipid complexes are then formed, which are returned to the cell surface. Thus, CD1 molecules acquire endocytosis lipid antigen during recycling of present and present these antigen without apparent processing. The natural killer T cell that recognize the lipid antigen may play a role in defense against microbes, especially Mycobacterium, which are rich in lipid components. Alpha delta cells are a small population of T cells that express antigen receptor protein that similar to but identical to those of CD4 and CD8 T cell. The alpha delta T cell recognize many different types of antigen, including some proteins and lipid, as well as small phosphorylated molecule and alkyl amine. These antigen are not displayed by MHC molecule, and alpha delta cell are not MHC restricted. It is not known if a particular cell type's antigen or antigen display system is required for presenting antigen to these cells. In looking here, we have immunodominance peptide protein antigen are processed to generate multiple peptides. So we see the internalization of antigen to antigen presenting cell epitope, then antigen processing. Processing generates multiple peptides which can bind to class 2 allele. Immunodominant peptide is taken. T cell respond to immunodominant peptide epitope. Yeah. So in summary, antigen antigen receptor of the most T cell recognize only peptide displayed by major histocompatibility complex, MHC molecule on the surface of antigen presenting cell. This is antigen presenting cell. CD4 helper T lymphocyte recognize antigen in association with a class 2 molecule, and CD8 cytotoxic T lymphocyte recognize antigen associated with class 1 MHC molecule. Antigen presenting cell captures protein antigen, process them, and display MHC associated peptide to T cell. Dendritic cells are the most efficient antigen presenting cell for initiating primary response by activating naive T cell, and macrophage and B lymphocytes present antigen to help a cell in the effector phase of the cell mediated immunity and in humoral immune response, respectively. All nucleated cells can present a class 1 associated peptide derived from cytosolic proteins such as a viral and tumor antigen to CD8 T cell. The Dendritic cell capture antigen from their site of the entry, usually through epithelial or production in tissue, and transport these antigen to secondary peripheral lymphoid organs. Naive T cells that recycle through these organs recognize the antigen, and primary immune response are induced in this organ. The MHC is a larger genetic region coding for highly polymorphic codominantly expressed class 1 and class 2 MHC molecule. Class 1 MHC molecule are composed of an alpha or heavy chain in a non-covalent complex with non-polymorphic peptide called beta-2 microglobulin. Class 2 molecule contain 2 MHC concorded polymorphic chains, an alpha and beta chain. Both classes of MHC molecule consist of an extracellular peptide binding cleft, a non-polymorphic immunoglobulin Ig-like region, transmembrane region, and cytoplasmic region. The peptide binding cleft of MHC molecule has alpha helical sides and 8 standard antiparallel B pleated sheet flow. The polymorphic residue of MHC molecules are localized to the peptide binding domain. The Ig-like domain of class 1 and class 2 MHC molecule can contain the binding site for T cell co-receptor CD8 and CD4 respectively. The function of class 1 and class 2 MHC molecule is to bind peptide antigen and display them for recognition by antigen-specific T lymphocyte. Peptide antigen associated with class 1 MHC molecule are recognized by CD8 T cell, where the class 2 MHC associated peptide antigen are recognized by CD4 T cell. MHC molecule bind only one peptide at a time. Every MHC molecule has a broad specificity for peptide and can bind multiple peptides that have common structural features such as anchor residue. The peptide binding cleft of class 1 MHC molecule can accommodate peptides that are 60 to 16 amino acid residue in length, whereas the cleft of class 2 molecules allow larger peptide up to 30 amino acid residue in length or more to bind. Some polymorphic MHC residue determine the binding specificity for peptide by forming a structure called pocket that interact with complementary residue of bound peptide called anchor residue. Other polymorphic MHC residue and some residue of peptide are not involved in peptide binding to MHC molecule, but instead form a structure recognized by T cell. Class 1 MHC molecule are expressed on all nucleated cells, where the class 2 MHC molecule are expressed mainly in specialized antigen-presenting cells such as the Dendritic cell, macrophage, and B lymphocyte, and few other cell types, including endothelial cells and thymic epithelial cell. The expression of MHC genes product is enhanced by inflammatory and immune stimuli, particularly cytokines such as interferon gamma, which stimulate the transcription of MHC gene. Antigen processing is the conversion of native protein into MHC-associated peptide. This process consists of the introduction of exogenous protein antigen into the vesicles of antigen-presenting cell or synthesis of antigen in cytosol to cytosol, the proteolytic degradation of this protein into peptide and binding of peptide to MHC molecule, and the display of peptide MHC complex on an antigen presenting surface of recognition by T cell. Thus, extracellular and intracellular protein are sampled by this antigen processing pathway, and peptides derived from both normal self protein and foreign protein are displayed by MHC molecule for surveillance by T lymphocyte. For the class 1 MHC pathway, protein antigen are degraded in proteasome, generating a peptide that bind to class 1 MHC molecule. Most of these antigen are synthesized in cytosol or introduced into the cytosol from the microbes or vesicles. These peptides are derived from the cytosol to endoplasmic reticulum by ATP-dependent transporter called a transporter associated with antigen protein TAP. Newly synthesized class 1 MHC beta 2 microglobin dimer in endoplasmic reticulum are associated with TAP containing peptide loading complex and receive peptide transported into the endoplasmic reticulum. Stable complex of class 1 MHC molecule with a bound peptide move out of endoplasmic reticulum through the Golgi complex to cell surface. Specialized antigen-presenting cell, mainly the dendritic cell, can ingest the virus-infected or tumor cell and transport their antigen into the cytosol for presentation by class 1 MHC molecule. This process, called cross-presentation, enabled the dendritic cell to initiate CD8 T cell response to antigen of ingested cell. For the class 2 MHC pathway, protein antigens are internalized into the endosomes, and these proteins are proteolytically cleaved by enzyme in lysosome and later endosomes. New synthesized class 2 MHC molecule associated with invariant chain are transported. Newly synthesized class 2 MHC molecules associated with the invariant chain are transported from the endoplasmic reticulum to the endosomal vesicles. Here, the invariant is proteolytically cleaved and a small peptide remnant of invariant called a clip is removed from peptide binding cleaved of MHC molecules by DM molecule. The peptide that were generated from extracellular protein then bind to the available cleft on the class 1, class 2 molecules, and trimetric complex class 2 MHC alpha and beta chains and peptide move to and display to the surface of the cell. The pathway of MHC restricted antigen presentation ensure that most of the body cells are screened for possible presence of foreign antigen. The pathway also ensures that protein from extracellular microbes preferentially generate a peptide bond to MHC2 class 2 MHC molecules for recognition by CD4 helper cell, which activate the effector mechanism that eliminates the extracellular antigen. Conversely, protein synthesized by intracellular cytosolic microbes generate a peptide from bond to class 2 molecules, class 1 MHC molecules for recognition by CD8 T cell, which function to eliminate cell harboring intercellular infection. The immunogenicity of foreign protein antigen depend on ability of antigen processing pathway to generate peptide from the protein that bind to self MHC molecule. Thank you for being with me. Now it was a little bit longer, but worth it. Here are some of the important link where you can find some of the important things that are happening with this thing that we have discussed. Thank you very much. Get prepared for this chapter that is following, chapter 7, the immune receptor and signal transduction. See you later.